Recording in progress.
We will start soon, and due to the pandemic, despite the pandemic, we managed to organize this conference. And today we have one day meeting about the intergovernmental cooperation. And we will talk about the related cooperations organized by the Ministry of Science and Technology. And this project is a bilateral. One party is EU. Yesterday, we received the notice that the name of this organization is changed to IEA, European Technology Agency. And actually, it is an EU project. And originally, the project is titled the FP, namely the Framework Plan of EU. And later on, it was changed to EU Project Plan 2020. And in this project, is an intergovernmental cooperation project organized by EU and China, and it is a flagship project. And it is a project in the food and agricultural and biotechnology area, which is a flagship project. So we know that this project is very important for China and the EU. So today, we have a one-day conference on this project. And this morning, the Chinese researchers will share their research results, and this afternoon, the speakers from EU will share their results. And this is the basic framework. And I will be the moderator for the morning session. Chris will be the moderator for the afternoon session. And besides China and EU, we also have friends from US. They will also present one report. And under the framework of the partnership, they participated in our conference. And the USP from two perspectives. The first perspective is under the framework of China-EU cooperation. And the second perspective is that we need to consider the time difference between China and the US. 1 week ago, we changed the sequence of our speakers. So our first speaker is Mr. Xie Zhuohong. He is a principal scientist at FCC, US Pharmacopoeia. And his topic is international collaboration to establish standards that combat food fraud and protect the food integrity. Can you hear me? I cannot share the screen now. Besides the characters in red, you can see share screen. In green, share screen in green. But I have no authority to share screen. Okay, it's okay now. Can you share screen, Kenny? Okay.
Thank you so much.
Now it's your turn.
Good morning, everyone, and thank you for coming to the meeting. My name is Kenny Xie. I'm the principal scientist at US Pharmacopoeia and Food Chemicals Codex. Sorry, I cannot visit you in person. I certainly miss Xiangshan Hongye. I heard now is the best time of the year to visit, but nevertheless, I am honored to give my talk at CIFSQ. So I am gonna talk about international collaboration to establish standards that combat food fraud and protect food integrity. I will first introduce Food Chemicals Codex and the background about why and how our standards developed. I will then talk about the international collaboration on the standards that we mentioned. So first, about Food Chemicals Codex, FCC. It is a compendium of internationally recognized standards for the identity and purity of the food ingredients. It was created by the US FDA and the US Institute of Medicine in 1966. It is currently published by USP, a nonprofit organization. And we have over 1,250 standards for additives, ingredients, and other food chemicals. Our standards are developed by our independent expert volunteers. We claim ourselves a fully independent source of food ingredient standards. An FCC standard consists of the following: the general information, which includes the chemical information and description. We also have the specification and methods that include the identification, assays, or the purity or potency, or impurities, including the heavy metals and those. Additionally, we have specific tests to provide additional tests to ensure the quality of the ingredients. In addition, we also have general tests and assays that provide test methods to users. I will introduce some of those to you today. So as we heard from the news, that we know supply chain is really, really complex. So producing those foods that are safe, nutritious, honest, requires control of and communication along the whole supply chain. Food supply chains are so complex, nonlinear, they are subject to sudden disruptions. This creates multiple opportunities for misunderstanding, miscommunication, mishandling, and food fraud and economically motivated adulteration. So what is food fraud or economically motivated adulteration, EMA? According to our FCC Appendix 18, we have the definition here as the fraudulent addition of non-authentic substances or removal or replacement of authentic substances without the purchaser's knowledge for the economic gain of the seller. There are two challenges to ensure our foods are safe and from food fraud. The first is that we are currently lacking in supply chain control. And the second is we don't have sufficient analytical testings, either that we do not test enough or we do not have sophisticated methods that are used. Standard methods are therefore needed to address those challenges. Not only USP FCC, but many organizations across the globe understand this need. It makes perfect sense to collaborate so we can share the knowledge and no one is reinventing the wheels. Through the Horizon 2020, USP FCC was able to expand the international collaboration on developing FCC standard methods to combat the food fraud. Experts from the members of Horizon 2020 participated in the planning, method development and validation, review and recommendation, as well as balloting. And among them are Dr. Wu Yongning, Dr. Wu Di, Dr. Huang Xiaoping, and Dr. Zhu Wei. They joined USP Ingredient Expert Committee, or the Dietary Protein Expert Panel, and contributed to the development of quite a few impactful standards. We really appreciate those efforts, and I'm going to introduce some of those outcomes. So I will just give a few examples on the standards that we collaborated. The first one is the amino acid profile for skim milk powder or non-fat dry milk. The principle is that the amino acids are the building blocks of the proteins. The proportion of the different amino acids varies between different proteins and can be considered as a characteristic of a given protein. Amino acid profile testing can be a useful screening tool to help substantiate the identity of a protein-rich ingredients. And on the other hand, that the typical total protein analysis by nitrogen determination methods would not detect adulteration on the addition or substitution with foreign proteins. We can obtain the amino acid profile by the following steps. First, we put the sample under hydrolysis by rapid microwave method. And we will use the AQC reagent to do derivatization. And after the derivatization, the amino acids can be separated through the UHPLC on a C18 column. And then using a UV detector at 260 nanometers, we can detect those peaks in the chromatogram. And after that, what's next is that we will compare the percentage of the total amount of amino acids between sample and the typical range to detect the potential adulteration. So how do we get a typical range of the skim milk powder? USP collected a library of skim milk powder and non-fat dry milk. 39 of them are used in this study. They come from different geographic origins, such as from the US, New Zealand, India, Ireland, Denmark, Argentina, and other places. And they are also processed under different conditions. 5 collaborative labs in the US and China participated in this study and contributed to producing those data. And here is the a sample chromatogram of the amino acids. Based on the performance, we selected 15 amino acids. That's based on the stability and also the position of those amino acids. And we calculate the amount of each individual amino acids in the sample taken as the percentage of the total amount of amino acids using the formula below. The typical range limits were calculated based on the following equation, which is somewhat similar to the student's T distribution, but being a bit more conservative. And here is the typical range of the amino acids profile in skim milk powder and non-fat dry milk. We also determined the amino acid profile using foreign proteins, which means the proteins other than the skim milk powder, and compare it to the skim milk powder itself. It can be seen that some foreign proteins have very different profile than what the skim milk powder has. Based on a series of spiking of different levels of those foreign proteins into skim milk powder, we can successfully determine spiking in the levels of as low as 0.1%, and some can be 1%. So for example, for soy and for pea, we can detect at 1%. And for rice protein, we can detect at 2%, at least 2% in spiking. And for whey protein, we can detect at 1.5%. And for fish gelatin, we can detect the adulteration at 0.3%. And for melamine and the wheat, we were not able to detect them through this study, at least to the concentration that we tested. And those are for the amino acid profile of skim milk powder and non-fat dry milk. And the other example I would like to present is the peptide identification of dietary proteins. The principle is that the proteins are digested by a specific protease, such as trypsin, and it will generate peptides, which are often characteristics of the protein used in the digestion. Peptides generated in this way can be detected and quantitated by our LC-MS system to provide species information on the tested protein ingredients. This can serve as a useful tool to help substantiate the identity of a protein ingredient. If expected peptides are missing or unexpected peptides are present, an investigation into mislabeling or an intentional adulteration should be performed. So the signature peptides can be obtained through the following steps. First, the samples will be extracted with 5 mole of urea in 50 millimole of cheese buffer. And then the extracts will be reduced with 1 mole of DTT and alkylation in 0.5 mole of the indole S-tamide. And following that will be a trypsin digestion, and then the extract will be subjected to LC-MS determination. Based on our literature research and our experimental trials, selected peptides and ionization conditions was determined, except for P, that each of the proteins have 3 peptides and 1 transition for each of the peptides. We use 2 system stability solutions to ensure the method works properly. Solution 1 includes a mixture of proteins at 20%. And solution 2 is a low-level solution that includes 0.1% each of the proteins in controlled matrix, which is the branch chain amino acids, BCAA. These 2 need to be run, and peptides need to be detected in both conditions. And the signals in the tested samples will be compared to that of the system suitability solution 2. The protein will be confirmed as present in the sample if its peak area ratio is higher than that of the system suitability solution 2 for all the peptide transitions described. So this method is established to detect proteins as adulterants in the authentic samples at the level of 0.1%.
那个张晶啊，我是张伟，我告诉你，我用我的这个
Sorry, by using Microsoft Edge, I don't have Zoom. I installed Zoom, and I think there is no issue. Hi. Can you use the chat function? It seems we have an outsider who is not going to speak at this session because now you are broadcasting, you are speaking loudly into the room.
The method is estimated to detect proteins as adulterants in authentic samples at level of 0.1%. And the 2 examples that I just explained were published in the FCC 12 in the 2nd supplement in March of 2021. Additionally, related to the protein ingredients, we also have a intact protein mass spec and the capillary gel electrophoresis. Those 2 are another 2 important methods that can detect the foreign protein adulteration. And the soy-lac hemoglobin, which is a niche protein ingredient that used in the Impossible Burger. It was also published in the FCC 12 third supplement. And in the coming publication, we are expected to publish a high impact protein called hemp seed protein that will be expected in March 1st, 2022. And finally, I will also want to mention our latest efforts on the food integrity, which are related to the standards we developed. We are currently developing a new type of analytical materials named FCC analytical materials. They are fit for purpose materials designed specifically for the food industry and can be used for method development, method verification, method transfer, method lifecycle management, method validation, or for other purposes. Some of the examples of the FCC, we call it FAMs, include the gluten in oat flour that was released. And we also have whey protein method development materials. Those are the whey proteins that will be useful for developing up to a state-of-art methods. And some of you may know that we also have matrix-based skimming powder materials that can be used for developing various types of methods, including the non-target methods against food fraud. And in my summary, that through the international collaboration, USP FCC develops methods, specifications, and materials to help guard our safe foods. And USP Food FCC provides various types of analytical materials, including the matrix-based reference materials for method development and validation. And these are all my presentations. And thank you so much for your attention. And feel free to let me know what questions that you have. And you can also feel free to email me at the address here. Thank you. Any questions, comments? Well, thank you so much. Let me just stop sharing and then, but you can still continue ask questions.
Please wait a minute because our moderator is giving a presentation next door.
那个非常抱歉，因为吴老师是一个大忙人，他现在
Dr. Wu is very busy, so he's giving a presentation next door. So I will be acting on his behalf as the moderator. I'm so sorry about that. We've had the first speaker. We'll go to the second speaker. This is Dr. Li Jiapeng, Assistant to the Director from China Meat Research Center. He will give us a presentation called Research on High Efficiency Detection Technologies and Meat Adulteration Risk Factors. Welcome. Hi, everybody, dear experts, colleagues, good morning. Today, I will talk about research on high efficiency detection technologies and meat adulteration risk factors. I will cover the 4 parts: technology background, research, application, and outlook. I don't know why this page turner is not working properly. Let's look at the background for meat adulteration, and this is economic-driven behavior, and it's a common problem at home and abroad. I've listed some of these common issues, both at home and abroad. This is going on over the years, and it's all over the country. For adulteration, in 2013, State Council have made the direction to combat any fraud of food adulteration. And this is also common overseas. In January 2013, there was horse meat scandal. In the US, Canada, Egypt, there are issues as well. From the data you could see, adulteration rate could be 20 to 68% with varying degrees. Regarding the horse meat scandal, the EU have carried out campaigns to deal with the problem to ensure authenticity. For the meat, there are risks as listed here.
There are 4 types of adulteries. First is the meat species adulteries. They use unqualified meat and mix them in the qualified meat. The second risk is the illegal usage of veterinary drugs. And the third risk is the mislabeling of expired date. And the fourth risk is origin counterfeiting. And there are different types. And the adulteration has strong concealment, wide range of hazards, and difficult to prevent and control. We needed to establish accurate, efficient, convenient, fast, and easy to use detection technologies and methods. We have a growing demand for this technology, and we need to obtain the sample information efficiently, and we need to get the information about whether the samples are adulterated and how serious is the adulteration. And the Horizon 2020 project is carried out, and we carried out systematic researches, and we established a detection technology system and R&D platform. I would like to share with you some of the technologies and methods. The first is the meat adulteration, and we established the multi-species synchronous detection technology based on multi-RT-PCR fusion curve. And we know that the fusion curve is a process of RT-PCR combined with the application thermodynamic analysis. And we also combine with the fluorescence information and the DNA spirals is combined with the fluorescence signals. And we also heat the samples, and in the heating process, the PCR products will increase from a low level to a high level, and the speed of the fluorescence wave will also change. And they are closely related to the thermodynamic characteristics of the RT-PCR. And there are, for the fluorescence wave curve are different for different samples. And through the multi-RT-PCR, we can have detection for different samples. And this method increases the information density, but the requirements for equipment and reagents have not been raised. Actually, the information density has a negative association with the difficulty of the equipment. And we need to select high quality species specific DNA sequence sites. And we should also optimize the response system and response conditions. In order to improve the efficiency of the design, we developed an intelligent primer design and R&D platform for multiplex PCR system based on Python. And the multiplex RT-PCR system and amplification conditions were optimized to improve the performance of the method. The platform is based on the digital assessment of the primers. Take specificity as an example. It is one of the key performance indexes of RT-PCR primers. And between the mismatch reaction and control reaction, delta CT were used as an indicator to construct a primer specificity prediction model by studying the occurrence law of non-specific amplification. And this is a model of the specificity. And in the heating process, the fracture is important. We can identify the qualitative relationship based on this platform and the model system and Python language. We established our method and process. First, we have the Python intelligent algorithm based on the model system and the DNA sequence information library. And then we explore the multiform thermodynamic targeting DNA, and then we design the primer. We detected the ingredients of 11 types of meat based on 6 RT-PCR detection. And for a PCR reaction can simultaneously detect six categories and more than 11 species of components from pigs, cattle, horses, etc. And the detection rate can reach 0.1 to 1%. And we can see the graphs of the detection curve from the peak, from the trend of the curve, we can get the result on the ingredients of the meat. And for different combinations of species, we established the multiplex RT-PCR. And with this multiplex RT-PCR method, we can identify the SNP positions with an identification accuracy of 94%. And we have an example of Qingyu pig. We established a RT-PCR model based on 9 core position points. And the 6 pigs appear in the 2 model systems. And if 6 pigs appear, then we can regard it as a Qingyu pig. If not, then it is not the Qingyu pig. And we established a series of methods for different combinations of species, including for fish, for other animals. And for the on-site detection, we introduced the portable infrared device, spectrometer device. And with this device, we realized the rapid on-site detection, and the spectral pre-processing is used, and the reduction algorithm is also combined in this model. And this model covered the adulteration of pig, cattle, sheep, chicken, and duck in pig, cattle, and mutton. Our model has rather good performance and stability and accuracy. And for the water injection, we also developed a method. Water injection in pork is very common in China, and we need to strengthen the regulation on water injection pork. However, the water injection methods are also updating, and there are 3 generations of water injection. The first generation is a simple water injection, direct water injection through water pipes before slaughter. The second generation is drug-assisted water injection and injection of adrenaline to promote the absorption of water by animals. The third generation is the compound drug injection to relieve the pain of the animals. In 2018, the Ministry of Agriculture issued the regulations on the quality and safety for agricultural products. According to the information of a risk exchange platform, market research and large sample analysis results, we identified the types of target drugs. And the animals may have some responses to water injection. So such target drugs can relieve the responses of the animals. And we also developed a high-performance HPLC-MS method capable of simultaneous qualitative and quantitative detection of the adulteration. And the water-soluble colloid was hydrolyzed into characteristics small molecules that could be detected by mass spectrometry by acid hydrolysis. The performance of this method is also very satisfactory. The above two methods are exported to Nestle in Europe in the forms of SOP and were widely applied. We established the identification technology of frozen meat storage time based on machine vision. We used the color space conversion, morphological segmentation, and color feature extraction technology to excavate the correlation between the characteristic mean value, redness difference, and frozen storage time, and the accuracy of detection for the meat with a storage time over 6 months can reach 96.3%. And the above technologies have been well applied, providing sample testing services and technical support for food regulatory agencies, public security departments, meat products enterprises, large supermarkets, e-commerce platforms, and consumers, and contributing to food safety supervision enterprises and supermarkets' internal quality control. And not only in China, but also in Europe, these technologies and methods are applied.
So these are some samples. We've undertaken some tasks from the government, and we've got certificates. And also we've got some requests to test samples for food sold online, and we've got certificates from enterprises. Technological outlook will make the technology system richer to meet different user scenario and demands. We'd like to establish a technological system with multi-technology dimensions, etc. We'll further expand the number of detection species, improve the quantitative accuracy, and reduce the detection cost. We'd like to construct the authenticity database of meat food ingredients. We've come up with regulatory recommendations better to improve the standardization system and optimize the standard development and review process, develop basic and performing parameters and requirements standards, and speed up the innovation. Adulteration is evolving as we are evolving our technology. This is attack and a counterattack. There will be new ways of fraudulent behaviors, new ways of adulteration, and we need to excel in innovation. We need to be innovative in methods, in building technologies, and we need to be faster than those bad guys. More innovative ways and technologies can be used to counterattack such illegal behaviors. And this is a process we need to push forward. This is our team. I thank my team members for their great contribution. With that, I thank you very much for listening. This is highly efficient methodologies, technologies for meat adulteration detection. Correct me if I'm wrong, and I'm happy to hear your comments or questions. Thank you. Any questions? We have used time efficiently. So there was short presentations in the first one and the second one as well. We can have a discussion. Do we have a microphone, or you need to step up here? Do we have a mobile microphone? Please identify yourself. I'm from Xinxiwang Liuhe factory. I'm in safety and quality management. We have pork brand, and in retail wholesale process, we found that some farmers just buy a few pigs from ours and also buy some other pigs in the name of our brand. So for such species, pig A, pig B, with the same kind of fodder, can we have differentiation? You could answer, and others could talk about this. This is about the different species of pigs. So in your company, what is the species or strain of your pigs, and what's the other fake species? I've talked about Qingyizhu, Lao pig, Beijing black pig. These are different strains. And also the normal white pigs. And if this is Yorkshire versus Yorkshire, it's not easy to identify the different species, but we can trace back to the individuals. It's like humans. Pigs have specific DNAs. If you trace back to individuals, for example, you use DNA fingerprint or gene fingerprint to trace back to individual pigs. If you trace back to individual pigs, the cost is a bit higher, but there's a way to deal with it. You don't have to test each and every pig. You can keep samples. For example, when you sell small piglet to the farmer, for each batch of piglets, you can keep samples. It's not really testing this, like blood, hair, etc., or some samples, and then you can trace it back. This is a physical code. You can have a biocode. That's the biomaterial you keep in the sample. If you suspect there is some fake pig in the name of your brand, you can compare those two samples from your original sample and their sample. And this is lowering cost, but this is for deterrence. So, Dr. Xing, I will talk about something related to this one, but I'm not so specific about your question. And in China-Europe cooperation program, we've seen some traceability study in Europe for pork. For this piglet, if it's from your factory, you know the information of the cell. If you want to ensure traceability, you can do that. Technically, it's feasible. So you can use the cell from your own factory, and this is the starting point for tracking the piglets. So it's better to trace. You see, for this cell, it could produce 200 piglets in her life, but it's not easy to find the information of the swine. I think in Europe, they are doing this kind of studies. Next. Through this program, we'd like to have digital DNA. I'd like to invite PhD Associate Professor of China Research Institute of SIQ to give us a presentation. Topic is application of digital DNA technology in food authenticity. Good morning, dear experts, colleagues. Good morning. I will give you a presentation. Is digital DNA and its use in food authenticity. And there was a question, and this question is related to what we do between China and Europe cooperation. I will cover a bigger topic. It's not about meat itself only. I will talk about what we've done in our team about digital DNA. And as the previous person asked a question, we can have some discussion. I will not go through the details. Dr. Li mentioned the current situation of adulteration in food is severe, and this happened in the past. And we've seen some news over the years. This happens in China as well as the world. Fraudulent behavior, adulteration is a global scourge. In 2018, from the FSA UK, they found 1 in 5 of the 665 treated samples, tested samples were partially or wholly made up of unspecified meat. We've used DNA barcode to have random sampling. We found around 23% of selected commercial samples were determined to be mislabeled. It's around 1/4. So this is getting serious. But in the meantime, there are food authenticity identification technologies. And in recent years, there's a new one. This is omics for food. This includes the proteomics, genomics, metabolics, and the non-destructive detection. So this is to deal with fraudulent behavior. By using genomics, we have DNA testing for this technology. You look at DNA nucleotides, and this is stable. Even if for the processed food, after they are being processed, DNA properties, the base pairs sequences will be maintained. And that's why this technology is used in processed food, and this is an advantage. For DNA barcoding, this is what we use in the program. In 2003, someone proposed this, and this is for categorization of biology. This is to copy the barcode in the products in supermarkets. So this is an ID number for the product. So this DNA barcoding leverage this concept. We can use several hundred base pairs to say this is the identity of the meat, and we call this DNA barcoding. This technology, this kind of sequence can represent the species, and this is a standardized way. And for this sequence, there should be enough variability. It is easy for application, and the DNA fragment is a bit short. That's the concept of the barcode. Here is the rationale. This DNA-based testing for each species, it will have its unique DNA sequence. This is the fundamental basis for DNA barcoding. For food authentication, you see, we are tracing the biological composition, and we're using this technology, we can test the composition and to see whether it is authentic or not. Why we choose DNA barcode? There are advantages with it. It's highly accurate. This is based on DNA sequences and voice morphological or proteomic and etc. Because DNA is more stable, others may not be so. So we can avoid the confounding factors from environment, from other proteomics, for example. This is based on DNA, and with this DNA sequence, we can build up databases which can be made open. Each and everyone can access and use it. So this could be a permanent database. Internationally, for DNA barcode databases, there are many of them out there. And in different species, there are specific databases. And there is the consortium for the barcode of life, and also the barcode of life, etc. So this is an international program or programs in categorization or identification of animals. This has become the gold standard. We need to sequence the DNA, so this relies on sequencing technologies. Sequencing technologies is developing by leaps and bounds. In first generation, we can have the lower Sanger sequencing, then next generation sequencing or single molecule sequencing. So owing to the advancement in these technologies, barcoding is getting more and more advanced and efficiency is up. Sequencing technologies are used in different fields, like in metagenomics, human genetic research, agricultural genomic, drug development, etc. In food authenticity study, sequencing technology is used only for a shorter period of time. Our team started to research into barcoding since 2010, and this technology is used in food authenticity. NGS barcode were used since 2016. In 2016, we checked literature, and we could find 2 or 3 papers. And at that time, DNA barcoding, high throughput sequencing were used for food authenticity testing. But in the current 2 years, you could see that very often.
And in DNA barcoding technology, our team has conducted a lot of work. For example, the species identification in meat products, the species identification in seafood, the species identification in spices, and in small berries and juices, and in poisonous mushrooms. And next, I would like to give you a brief introduction. First, the animal species identification in food products. And this is the work we carried out earliest in 2008. We issued the national standard for the animal species identification in food products by barcoding technology, and for 50 ingredients in animal species, we set standards for identification. This standard is to identify a single species of the food products, and based on the previous technology, we developed the second generation with high throughput, and we will identify and analyze all the ingredients of this food. And this chart shows the workflow. For the food species, we first have a PCR expansion, and then characterize the different ingredients and indicate with marks so that we will accurately know the ingredients of each species. This method can help us get the information of the ingredients of the animal source food. It's quite valuable work. And we also used our method to identify the commercial food products. For example, the camel meat. We used the DNA barcoding technology to identify the ingredients of the camel meat. However, from the camel meat, we only detected mutton and beef. And we can also identify the sea cucumber products. And actually, the sea cucumber is widely spread across the world, but it is not consumed in every country. And we heard Liao cucumber, and there are over 1,700 types of sea cucumbers, and but only 600 categories have economic values, and the European countries and the US, they do not consume the cucumbers. We collected some cucumbers from the market. Actually, they have different shapes with what we usually see. Their forms, the sizes and the colors of these sea cucumbers are different, but they are edible. Of course, their taste and the texture of meat are different, but their prices differ due to their different origins. And the prices of the sea cucumbers are usually high. And besides the dry sea cucumbers, we also have the byproducts, and the alteration in such byproducts are even higher. For the dry sea cucumbers, maybe there is mislabeling. Maybe the low quality sea cucumbers is adulterated into the high quality sea cucumbers, and with the DNA barcoding technology, we can identify. We used 2 generations of the sequencing. One is the basic sequencing, the other is the high throughput sequencing. We developed 2 systems for the sea cucumber itself and for the products of sea cucumber, respectively. These are some examples of the species identification of sea cucumbers based on our technology. these dry sea cucumbers. Actually, we can see their original shape. Maybe the labels on the sea cucumbers products are false. They use the mislabeling to improve the price of the product, but for ordinary consumers, they cannot identify. And for the products based on sea cucumbers, they are highly processed. And according to our identification, we found that they contain more than one sea cucumber species. These are some examples of the highly processed sea cucumber products. And we also use our method to identify the species of the dietary spices. We can identify the species of single spices or compound spices. The 5 spices in China is a traditional blend of ingredients balancing 5 basic tastes: sweet, sour, salty, bitter, and umami. And the ingredients are quite complex. We developed the DNA barcodes for selection and validation. And with our sequencing, high throughput sequencing technology, we developed a complete method. And after that, we detected some dietary spices on the market. Some spices are mislabeled, and some spice powder also contains corn or grass ingredients. Through the high throughput identification method, we can have very accurate qualitative identification. And we also used our method to identify the poisonous mushrooms. Poisonous mushroom is a very common issue, and the DNA sequencing method is very suitable to identify the poisonous mushrooms. For a certain kind of mushroom with a high poison, we developed a specific DNA sequencing method, because for different mushrooms, for different species, they are suitable for different DNA sequencing methods. And we needed to explore and screen a specific method for a specific species. And we needed to develop a certain method and develop the primer. And we used our self-developed DNA barcoding technology to identify the Amanita. And upon the development of this method, we also used our technology to identify plants. So we developed a website for the barcode. And on the website, we can see the morphological descriptions of the plant. And we established a small database with information uploaded to the website. And we have for computer and mobile and we provide a QR code. Everyone can get information of the species upon scanning the QR code. And it is open source material available for the public. This is a website developed by our team in recent years. And it is quite effective and useful. In the future, it can cooperate with the regulators and better be applied. Just now, I only introduced you the technology itself and how to transform the biological information to our database. And in our institute, we also have traceability system. For example, we developed a traceability system for bird nests. For every bird nest imported from foreign countries, we will put a QR code so that the consumers can scan the QR code to get the information of the bird nest. And we combine the physical traceability and the biological traceability, so it will effectively prevent the adulteration. And it is the prospect for our future application, and the big data blockchain technologies can also be combined. I will update our research results. Thank you for your attention.
Any questions, please? You may ask Dr. Xing. We have young researchers and we have young minds. Do you have questions, please? Seems you are too shy. In terms of traceability, we'll finish the first part. We have two other speakers on safety. The first speaker is Jing Zhang, a research fellow from Beijing Center for Disease Prevention and Control. And this is the deputy director of the lab. You see, he's from CDC Beijing, and in his community, there is someone with COVID-19 positivity, and he's banned from coming here. He will give us a presentation online, and he will be paying attention to perchlorate, which is an important contaminant. There are many risk assessment, validation. It is approved that there is a committee which reviewed this chemical method, and this analytical method has become part of the draft for national standard methods. So his topic is occurrence of perchlorate in foodstuffs, analytical methodology development, and the Chinese side to exposure. Zhang Jing. Dr. Wu, hi. The floor is given to you. you can go on with your discussion until 10 past 10. Okay, dear professors, colleagues, thank you, Dr. Wu, for introducing me. Because of COVID-19 cases, I cannot make it here. Sorry, I have to give you a presentation online. Yeah. Okay. We have to do it online. I will share you what we have done in our lab. My topics, occurrence of chlorate in foodstuffs, and also the methodology development and Chinese dietary exposure. I cover the following topics on my agenda: background first, analytical method and development, and dietary exposure analysis and summary. First of all, let's look at the background. For these perchlorate and chlorate materials, they are important and they are ion forms in terms of contaminants. For hypochlorite, this is from the processing from the nature. For chlorate is for disinfectant. However, for chlorate and chlorate, they have impacts on the thyroid. They will be competing against iodine. So it will jeopardize thyroids. As it is high oxidative capability, it could cause damage to red blood cells. So, for such contaminants, they are of great concern in different fields. There could be dietary exposure. It could enter the human body through food or other means for susceptible people like infants, pregnant women, or people with thyroid dysfunctions. They will have bigger impact. Therefore, based on its toxic effects, EU in last year have declared the mandatory limits. For chlorate, there is regulation 2026A5. For perchlorate, the limit is 0.01 to 0.75 mg per kg. 0.01 is for infant food, 0.75 is a bigger one. It's for tea and herbs. For chlorate, there is 2020-749 from EU regulation. The limits are 0.05 to 0.7 mg per kg. According to this directive, this is for fresh food, like fruits, vegetables, etc. Because of such limits, we'll see whether the food meet the criteria. There should be a basis to determining that. We need to have analytical methods, and this is up to the researchers like us. Up until now, for chlorate and chlorate detection, if you look at the food industry, we use ion chromatography or IC coupled with tandem MS or HPLC-MS-MS. For HPLC-MS-MS, this is ion exchange columns, and the use of such columns will have issues in short service life. There are complicated mechanisms in food when we use ion columns or HPLC-MS columns. There will be limits in sensitivity. There will be higher costs. And as I mentioned earlier, the service life of such columns are short. And in operation, there will be some complicated operations. So in our lab, we try to optimize such methodology. We used LC-MS-MS methodology. First of all, for the MS methodology, we use this water UPLC. This is to look at percolorate and colorate MS conditions. This includes the parent ion, product ion. This is about their voltage and the energy. We'll use isotope for dilution. So for oxygen 18, we've optimized the parameters. Next up, for the LC column, I will give you some updates about what we have done. For percolorate and colorate, these are high polarity by using LC. You must use a column with high specificity. We've chosen several types. Trinity P1 is an example for the filling material. On the nanospheres, we have particles. In the inner walls, there is exchange of negative ions. There is reverse conservation. So this is a column with different mechanisms. By using this column, we use the ACN and NH4FA. In the right lower corner, we've shown the peaks of those two ions. Comparatively speaking, you can get a very satisfactory result. By using such means, we've tested several types of columns. And as I mentioned, there is Trinity P1, or Halic matrix is water's porous DEA. On the silicon, there is DEA radical groups, and this is to preserve the high polarity materials. And we use ACN and AH4FA. We've used Shishindou's Halic PC. This is also Halic matrix. On the surface coating is acetylcholine. By using choline radical groups, it could keep the colorate ions. We tried water's CSH PFP. And for this one, this is reverse column. On the surface, they've added a positive charged ion. And we use ACN and 0.1% of FA. For these columns, we've had comparisons. If you look at the peaks, Trinity P1, Halic PC have wider peaks. From these columns, if you process the real samples, we found some issues. Normally, for the preservation of colorate ions, there will be a wider peak or deformity of the peak. So at the end of the day, we use water's CSH PFP, which can ensure the best outcome. So at the end of the day, we've selected the CSH PFP column. There is positive charge, and this is methyl and acids. So compared with other systems, we don't use buffer salt here. And for the user, this is more user-friendly, it's simple to use. And for such systems, in regeneration of the column, the operation is very simple. If you look at the peaks of this one, and it has a narrower peak and it's like a spike. Then, for different food, we've had some optimization. We have optimized the extraction conditions. We've compared with water and the methyl, etc., and we've compared them accordingly. For colorate and percolorate is ions with high polarity. It's easy to be dissolved in water. But if we use water only for extraction, it will be turbid, like especially for milk powder or egg-based material. We need to remove the impurities. If we compare methyl and the other one, this acetonitrile can have better results. And in this system, if we add some formic acid, it will have better sedimentation. After extraction, we have checked and optimized the purification process. Normally, in purification, we use solid extraction or dispersed extraction because we're developing the standard methods. We use the solid extraction method, which is highly repeatable. In this, for the extraction columns, we've had tests. This includes carbon 18, prime HLB, prime HLB, HLB, WCX, MCX, and DCB. I've listed them. We use spinach and also a composite spice, and we've compared the purification effects of those different columns. If you look at the appearance, if we use other purification columns, if you look at the pigment side, there are some traces, but if you use DCB, it's more clear. And from removal of pigments, DCB is the best one. So we could use DCB to purify the matrix of food. DCB in literature is mentioned, and it's commonly used for purifying the matrix in testing percolorate.
For the high salt spice, and if we only use the GCB for detection, we identified some problems. For example, for the sauce, we identified that the peaks of colorate and percolorate disappeared, and we are considering whether the source sample are high salt containing samples with a concentration of salt over 10%. So the chloride ion has some impact on the peak form. Therefore, we used the standard solution of chloride and perchlorate, and add sodium solution. When the sodium chloride has a concentration of 500 mmP, then there is a peak for chloride. And when the concentration reaches 5,000 mmP, we can see the response value of the chloride and the perchlorate only reaches 1/10 of the previous value. So when we test the high salt samples, we needed to further remove the salt and purify the extraction liquid. And for the salt column, it can remove the chloride ion in the solution. So we use the two columns and compare them. And we found that no matter for the source sample or the oil sample, the purification of the silver hydrogen column has a better effect. And the purified liquid is cleaner. And finally, we get such a purification process. For the fresh samples and the drinking, we will take 2 grams. But for dry samples, for example, powder or cereals or crops, we just take 1 gram and we use 4 milliliter 1% FA water and 6 milliliter ACN for extraction. And we purify the sample by GCB cartridge. And if it is a high salt sample, we use AGH for further purification. And for this method, it can reach a very wide range, linear range for perchlorate. The range can reach 100 pp per milligram, and for chlorate, it can reach 10 to 1,000, so that we can ensure that the external interference will be minimized. And we also validated the methodologies in our laboratory. And almost for all the columns, the value can reach such a range. And for chlorate, the detection level can reach 0.5. And for perchlorate, it can reach 0.5 milligram per kilogram, and for chlorate, it can reach 5 milligram per kilogram. The recovery rate can reach 82.3 to 109.6%, and the accuracy can reach a RSD lower than 15%. And this method is suitable for chlorate and perchlorate determination in a variety of food samples, including animal producing foods, cereals, vegetables, fruits, drinking, dairy products, and seasoning samples. And on Monday, I surfed on the internet and found this method available for the public. I hope that the experts can provide comments and suggestions for our method. With this method, we detected many samples, and here I just give you an example of the seasoning samples. And we tested 84 samples. The samples include 13 soy sauce, 13 vinegars, 36 spices, and 22 other species, and we have a high detection rate and detection level of the chlorate and perchlorate, especially for the spices. The highest detection concentration can reach over 10 ppm of perchlorate. And the average detection rate of chlorate can reach 1 ppm. According to EU standard, the limit for the chlorate and perchlorate concentration in spice is 0.5% ppm at the maximum. So we need to pay attention to this issue. And we found that the pepper sample has the highest concentration. And for the pepper sample, its concentration of chlorate and perchlorate is far higher than other dry spices. And we will explore why the pepper has such a high concentration of chlorate. After establishing our method, we hope to understand the intake level of the population, and the dietary intake is a major source of the contaminants exposure for humans. Total dietary study, TDS, in short, is one of the most effective methods to assess the potential health risk to the general population posed by food contamination. And according to the 5th and 6th TDS, we collected the samples and analyzed the content of the perchlorate. The 5th TDS was conducted from 2010 to 2012, covering 20 provinces, covering 70% of the Chinese population. And based on that basis, we added 4 provinces. So the 6th Chinese TDS was conducted between 2016 and 2018, covering 24 provinces. For a specific province or region, we will select 3 sites. Either a city or county, and then conduct the cluster analysis, and we will make a mixed sample for a certain province. Finally, for each province, we will obtain 12 categories of samples, including beans, crops, cereals, etc. And finally, for the fifth TDS, we collected 240 composite samples, and for the sixth TDS, we collected 288 composite samples. And next, I would like to give you the example of the sixth TDS. We have a total of 288 samples. There are 273 of them detected containing perchlorate with a concentration range of 0.5 to 129 milligram per kilogram, with the highest level occurred in vegetable sample from Guizhou province. Vegetables and eggs have a high detection level of perchlorate, and for drinking and cereals. Iodine has a high detection rate. According to the sixth TDS, Jilin province has the highest detection level of perchlorate. The maximum perchlorate level ranged between 0.51 and 41 milligram per kilogram, followed by Hebei province and Jiangxi province. We used the iodine assessment method to calculate the dietary exposure in China. For a certain sample, the concentration of iodine of this species is multiplied with a consumption amount, and also consider the weight of the human body weight. The detection rate is rather high, so we use a half MLOD.
See this one? We've checked the value of dietary intake, and we made this color map in China. For the fifth TDS in Qinghai province and the other two provinces, the volume is above 0.7 microgram per kg. And in the sixth TDS, we found 5 provinces where this is above the threshold value. 0.7 microgram kg per body weight per day. It's recommended by National Academy of Science of US and EPA. This is the threshold value for maximum uptake for adults. So hyperchlorate exposure in China should be something we pay attention to closely. And if you look at the exposure in China and you compare that with countries' literature from other regions, you could see ours is at a higher level. In the US, they've had a survey So it's from 08 to 12, and data was published in 2019. Sorry, 2018. And this is for the whole population study. It's 0.13. And JECFA gave a recommended value is 0.12. In France, they've used less variety of food is 0.08. In Korea, in 2012, based on massive sample testing, it's not for the whole diet study. We've got a lower value is 0.04. In Taiwan region of China, in 2019, they published a paper, and it was quite high. This is higher than the number from the US. And this is maximum allowable intake. It's about 1.06 microgram per kg body weight per day. In the fourth TDS study, it's 0.68 unit in China. And we've had fifth and the sixth TDS. The average was 0.35 and 0.45. From this, you could see, compared with the fourth TDS, we have lower numbers now. But in the sixth TDS, it is a little bit of a pickup compared with the fifth. If you look at the exposure, this is higher than the recommended threshold from EU. In EFSA TDI, this is 0.3% unit. And this level is lower than the threshold from EPA US. Anyway, our level is higher than the Western counterparts. Mainland China and Taiwan are similar because we have similar dietary structures. If you look at the contribution from major food for hypochlorite, whether it is the fifth TDS or sixth TDS, you could see vegetables contribute the most. It's around 50% in the fifth. It's 46.9%. In the sixth, it's 45.9%. Then it's beverages. Then it's cereals. In those two studies, there was a gap about 6 to 8 years among the two sessions, but hypochlorite remain high and the contributing factors remain the same. If we'd like to control the exposure of hypochlorite, we need to consider vegetables, beverages, drinking water, cereals. We need to manage them. Okay, let me sum it up. First of all, we've developed analytical methods for simultaneous determination of chlorate and chlorate in a variety of dietary samples. We've used SPE, purification, LC-MS analysis. Furthermore, we have established such method. It was used in real samples. Seasoning products presented a notable contamination of such chlorate or perchlorates. We've had the fifth and sixth TDS in China, and we've calculated the values. These are the average intake of perchlorate of Chinese adults. It was 0.35, 0.45 microgram per kg body weight per day. This is higher than the recommended value from EFSA. The maximum daily intake is 0.3 in EU. We need to pay attention to that. That's all from me. Thank you all very much for listening. Thank you, dear moderator. Thank you. You have delayed our schedule by 15 minutes. But let me correct one thing. In Taiwan, China, if you follow JECFA, TDI, it's not above the threshold value. If you follow EU method, sometimes it's higher than threshold. According to US EPA, there is a committee of risk assessment of China. We have similar levels with the American TDI. For the diet, for the risk assessment, it's not above the counterparts. When we use JECFA, we need to consider the conditions for infants and neonates. So we need to look at the healthy outcomes. We use Noel. It's not about 0.5 thyroid outcome. So in JECFA, it's 50% of the thyroid. They say there is a compensation capability, but we use Noel. So the maximum dose without effects. This is to improve the safety index. That's why we use 0.7. We have 10-minute break. Please come back at 10:30. Thank you, Dr. Wu. You come earlier than me. You were on time. So we'll have Li Bin, a deputy researcher, associate researcher. So this is 2020. So what's important here? We need to have this reference lab, and more importantly, in EU, they have a new method for detecting dioxin, and efficacy and validity is not so good. So in China, we've had intra-lab validation. So we'll invite Dr. Liu Bin from China National Center for Food Safety Risk Assessment. She'll talk about validation of GCMS, MS, analysis of dioxins and dioxin-like polychlorobiphenyls in food. Good morning, dear professors, colleagues. I will talk about this topic. It's about our validation. Dr. Wu has introduced what we have done. This is part of the EU-China CIF. So this is about RL 2020. GCMS have been recommended from EU, and in our side, we'll look at its validity, look at the parameters. We've had comparisons in reference lab in China, and as well as EU National Reference Lab. And ultimately, we'd like to convert this into a Chinese national standard for detecting dioxin. So this is to supplement the original methods we had, and I will go through that with you. For dioxins is a contaminant in natural environment. This comes from the incineration of wastes, and also volcanoes bring contaminants as well. And this has accumulation, and lasting period is long. In the food chain, it is accumulated, and you could see the exposure in China. You can see 95% is from food. Dioxin detection in food is something important. For detection of dioxin in food, we use MS, and this is magnetic, and it is of a higher cost, and it needs great operators. So we are trying to see whether we have alternative methods. The EU use GCMS, and you can see since 2014, there was the law. GCMS was recommended to test dioxin in food and animal fodder, and there was some improvement since then. According to this law, GCMS should be used is to validate, and it's a confirmatory method for exposure test and trend analysis of concentration. We use the magnetic MS. In the EU law, GCMS analytical methods have been required in technical aspects. If you meet their requirements, then you can use the method. There are several aspects to it. According to this method, you have to reach 1/5 of the maximum level. There are 4 technical aspects. In dioxin, there are 6 chlorine furans, and this overlap should be less than 25% in peak-to-peak. And also, when you look at the parameters of MS, when you look at that, the parent ion should be a different one, and they should have their own product ion. Number 3, whether it's standard curve or otherwise, the relative ion intensity should be minus plus 15% for selected transition product ions, and also resolution of each should be higher than the unit mass resolution. So in our lab, we validate such methods. We've used some the devices, 3 of them. 2 have been optimized. One is APGC, and we've had validation on these 2 kinds of sources. These are the parameters of the method. The method need to consider this as part of the supplementing method for the national standard. So we refer to the parameters of the original methods and has some updates, and we optimize the parameters. And this is this one for GC, similar to the old national standard for resolution. It's now updated. So this is the score of dioxin and PCCDs. So this is the information and 2 parent ions and also product ions. In the 3 machines, we've had TIC maps. We've chosen 3 different concentration levels. And this is quite low in this map. I'm not showing the differences. I'm showing we are using samples of different concentrations. For this part, I mentioned point 1. This is about the degree of separation, whether it is furan or dioxin. We've had comparisons. At extreme lower concentration, for 20 microgram, you can see there is good separation. This is ion ratio, and we've had validation. In standard curve, we've got 7 points. We've had comparisons. You could see in all the dioxin, the fluctuation is minus plus 15%. This meets the requirement from the EU. On the basis of that, we've had validation on the 4 parameters, and we try to see the sensitivity of our instrument. We've listed the IOQ. We have lower concentration. We tested the equipment and its detectability and stability in detecting lower concentration. So 2378 CDD 0.01. And you can see the response of the machine meet the requirements of testing. Then in the national standard, when we have tests, we use the entry barrier for any machines. If you want to do the test, you have to meet this criteria.
A device should meet the requirement. Similarly, at this concentration, we also validated its sensitivity, and we have for interday and intraday validation to test the instrument stability. And how to identify the ILQ? Because the dioxin is a very special material. So in determining IOQ, we have some confusions. And we validated the European method and adopted the IOQ determination method recommended by EU. And the lowest concentration point on the calibration curve that gives an acceptable and consistent deviation. And this concentration point is acceptable for our device. In combination of the curve of this method and under the lowest concentration point, the deviation rate is below 30%, so it can meet the EU standard. After determining the quantitation line, we will see whether it can meet the requirement of EU. EQ can reach 0.06, which can meet the EU requirement. And the above analysis is the validation on the European Union method, and we want to transform the method into our national standard. So for the precision, accuracy, and the comparison with the analysis result of the national standard, I would like to introduce you in the following part. First is an experiment on the precision. We selected 3 samples and we carried out 5 detections for the intraday precision and carried out the interday precision for 3 days. And we will obtain the result of TEQ for PCDD and for PCB. And the level is below 10%. And for PCB, the concentration is rather low, lower than the threshold. And for other parts, the result can meet the detection requirement. And in terms of accuracy, we selected 6 different substrates of inter-laboratory comparison study samples. And 4 categories of samples were tested, including beef, fish, salmon, herring, and the TQ is ranged between 0.054 to 0.3. And for cheese, the value is slightly higher than 2. So the reference value for cheese has a gap with the threshold. So in a low concentration rate, there is some deviation, but normally it can have a high frequency accuracy. And the next part is a method comparison, because we want to determine the national standard, so we need to confirm whether it can meet the requirement. And we have 7 parallel testings and have the standard deviation analysis for 2 of the results. And the result of the standard deviation is higher than 0.05, so there is no significant difference. And just now, it showed the result for dioxin, and this is the result for PCB. And the next part is the interlaboratory validation. we selected 5 qualified laboratories and sent some samples to them so that they will test the linear range, LOQ, precision, etc. And this is a result of the standard curve. And the linearity can meet the requirement. In terms of the interlaboratory validation, we need to pay attention that because the dioxin has some chemicals with very low concentration, and with such a low concentration, we should not use the RSE method lower than 20. So we used the PRSD method, and we have some calculation. As long as the PRSD is lower than the required concentration, for example, lower than 45%, 63%, 89%, then it can reach the requirement. As we can see, we used 4 categories of materials. And on the left side, we can see the standard values of the 4 categories of the materials. And on the right side, we can see the deviation. And we can see that it can meet the comparison requirement. And we also have the comparison between mass spectrometry and GMS. And we should also treat the values. So this is the processed value. And the above is about the validation of the national standard. We successfully completed the development of the national standard and also had some revision and update. And now the national standard for food safety is open to solicit your opinions. If you have any comments or suggestions for this standard, you can log on the website and submit your opinions. The next part is another part of our project, namely the EU-China interlaboratory validation, and we obtained 6 groups of samples, and we sent the samples to 6 laboratories in EU and China respectively and compare the results. But we cannot analyze the standard deviation or the significant difference. So we have some linear comparison of the data and also draw the column chart. Our experiment is affected by the COVID-19 pandemic. In the future, we hope to obtain more data, more samples, and have more accurate comparison. FARA provided the samples, but after the pandemic, it's difficult for them to send the samples to us. Based on this result, although we encountered difficulties in samples, yet we have realized the data sharing on the platform. And based on our project, we created a platform for data sharing. We hope that while we share data, we also hope to share our experimental instruments. We will see the parameters, the detection methods, risks, and other situations. So our platform can enable us to share data and methods. We hope that more information, more data will be shared on the platform. And we hope to achieve mutual recognition of the data between China and EU. And finally, I have a summary of our work for the EU method and the GCMSMS method. We have conducted the validation. And we can see that our method has no significant statistical difference with the European method. And according to the comparison result, we are quite satisfactory of our for our method. Now, our national standard is in the process of opinion solicitation. This is my presentation. Thank you. Thank you, Madam Lu Bing, for your excellent presentation. And in the whole process, we have two methods. One is a colorate, the other is dioxin. And now the National Health Commission is soliciting opinions from the public. Therefore, we need to develop a number of methodologies. Nitrofuran has 4 products, but now we have 8 products for nitrofurans. So we must detect the metabolic products of nitrofurans. So we need some methodologies. So our next speaker is Yu Xuezhi, Associate Professor from College of Veterinary Medicine, China Agricultural University. He will give us a presentation on hapten design and synthesis, monoclonal antibody production, and immunoassays development for nitrofurans.
From antigen design and MAB generation and method. So this is our part now, Professor Yu. What's wrong? We try to show the slides. Now it's coming. Hi, morning. This is Yu Xuezhi from CAU. Great to be here. My topic is hapten design synthesis, monoclonal antibody production, amino acid development for natural furans. This is my agenda. I will go through background, content, results, analysis, conclusion, outcomes. Dr. Wu talked about this background. So this is WP4 of the China-EU Food Safety Cooperation H2020 EU-China Safety Project. We make antibodies and have amino acids developed here on our part. For natural flavonoids are class of synthetic broad-spectrum antimicrobial drugs, which used in livestock, bee, and agriculture. The metabolites are carcinogenic. In 1995, it was banned to be used in food, but natural furans have great efficacy, lower cost. So in the livestock companies, they still use it. And there are some illegal use of such antibiotics. In the market, we have the four major parts like nifurosol, natrovin, nifurozatide, etc. I will go through another four. You see, a lot of these are widely used and their metabolites jeopardize human health. But we are looking at the rapid test. But if you look at the rapid test, there are few assays, and such natural furans are dangerous. And we need to develop antibodies and also rapid test methods. Now, there's only antibody against natural furosol, and it's not the best. But for the other three, we don't see antibodies yet. You see in the market for the traditional four, if you look at the aquatic culture, this is taking a high proportion of the drugs they use. On the left, this is the original material. On the right is the possible metabolites. They are of special structure. They have smaller weight. HBD is low in molecular weight, and HGD is 74 in molecular weight. It's very difficult to test. We can look at the revitalization products of these. Whether it's rapid test, you need to have the revitalization. And for the rapid tests, even if it's a card, you use a card and run it. But for the pre-treatment, you need to have the revitalization. So when we tackled this topic, we thought about it. Can we make an antibody? We don't go through the revitalization. We can identify the four metabolites. With that question in mind, we started our project. And if this is direct way to test metabolites and eliminate the revitalization, then we need to have our original designs. First of all, for nifurozatide, look at the HBD, it's 152 in molecular weight, it's quite small. And to make antibodies, to identify HBD without revitalization, then we have to preserve the structure of HBD. In the four antibodies on the right, we need to have this arm, and the HBD part is exposed into the immune system of mice. When we design the antigens, aptogen, we look at the structure size, spatial configuration, electronic configuration, and hydrophobicity. We did not touch upon this HBD, and we just try to connect with arms. There are purposes to establish spacer arm to ensure there's better conjugation with the carrier protein. And this is away from the carrier protein. When their conjugation is too near to the protein, is not exposed or is enclosed by a carrier protein. So having this spacer arm is very important. We've analyzed the structure. We've designed 4 spacer arms. But according to our speculation, this is of small molecular structure, simple. If we use linear aliphatic spacer arm, is hapten B, gonna lead to immune response. So we need to introduce a bigger spacer arm. We've designed 4 haptogen and are they appropriate? We checked the electric charge. We see whether this is an appropriate design. Look at picture A. For the hapten and HBD part, this is overlapped. So in HBD carbon 11, we can introduce the arm will not jeopardize the structure. This is HBD part and haptens are fused. So introducing this arm does not jeopardize HBD. We should not damage HBD. From panel B, you could see we've introduced different spacer arms. HBD charges do not change significantly. This is the color bars. After analysis, we find hapten C. Its charge distribution is the most similar to HBD. So before any processing, hapten C can be the best one. Is it true? We need to test it. Hapten C is the best by looking at the conformational parts and also charge analysis. We found the 4 can be both used to make antibodies. First of all, we've synthesized the haptens, and we're afraid of lack of identification. We're afraid of metabolites or privatization. There's A, B, C, D, and E is the phosphorylated result. We've had MS studies. We found the 5 haptens were made successfully. And for the 5 structures, we've had an MR, and it shows that we have synthesized them successfully. For the 4 haptens, we have coupling with the carrier protein. There was successful conjugation and blank BSA, and the 4 antigens we've had a Molotov processing and A, BSA, and the other 4 are synthetic antigens. So hapten BSA were 11.4 versus 1, then 14.4 versus 1, and 8.2 versus 1, and 10.4 versus 1. This is successful. And these were used for mice. We've inoculated 5 mice in each group. After this, we can see the most appropriate one happens in mice 5 in hapten C. This is in line with our speculation. After inoculation, this result is the best in mice 5. This is for cell fusion to make MABs. In mice 5, we've got 4 MABs. 2F12 is better, but for the other 3 strains are okay. Hapten B is the best one. And for the IC50, it's 0.25 and 0.10 ng. For 2F12, we've had a specific analysis. We've used different metabolites, derived products, and other natural furans. You can see HBD, NPH, BD, And nalfurazide has very good specificity. It could identify HBD. It could detect metabolites. It can avoid derivatization, and this was published in Food Chemistry. Based on that, the antibody of 12, actually there are 4 of them. We've had optimization. 12 is the best one. Based on this antibody, we've developed the ELISA kit. The best condition for operation is 37 degrees Celsius, pH 7.4, iron concentration less than 0.1 mole, and there should be no organic reagents. You can see this 0.25 in IC50. And in chicken meat, we've found this is applicable to detect the nalfurazide metabolites in chicken. We've added 0.5, 1.0, 2.5 ppb of HBD. You can see the recoveries could be 98.5, 99.4, or 5.6%, and the CV value is quite good. For the kit, we need to look at stability and storage. We've had a stability test. The kit can be stored for a year. So there was for one nalfurazide, and we've tested the others. I show you part of the results. The second one is nalfurosol. DNSH is the metabolite. Molecular weight compared with the HBD, this one is bigger. And we've tested the structure of metabolites, and we've designed 5 heptadines, and we've had successful heptadines after conjugation. This is used for the immunization of mice. After testing, heptadine E is the best one, and still mice 5. And there's high sensitivity. We use heptadine E5 for fusion of cell fusion. Again, we've got 4 strains of MABs. The optimized one is 3H10, and heptadine E is the best one. For the metabolites and derivatives is IC50 at 0.91, 0.60. The previous one. Again, we've checked the specificity. This antibody we made have very good specificity. It identifies the metabolites, derivatives, and nalfurosol. So it has very good specificity. And this can be used to detect the metabolites rather than the derivatives. Then Natrovin. Compared with the previous 2, AGD is 74. It's too small. And if you want to make antibodies for this one, it's quite difficult. When we started this, we did not feel sure. We've made another one. It's for poison of rats. It was 70 and sensitivity was not good.
And we also designed the derivatization structure. But now we only made 2 immune source. And now we have only data for the 2 immune sources, but for other sources, the postgraduate students who conducting the related experiment. And according to this experiment, we found that AGDB3 mice was selected for cell fusion due to high sensitivity towards the AGD and the derivative. And we obtained 2 MABs from the 2 mice. The 4G8 has a better sensitivity, and the effect is rather good, and the sensitivity is below 1 PB. And the fourth is the nephrodizone, and the molecular size is very small. It's only 103.8, and we also designed the derivatization. And actually, the sensitivity for 2 mice is very high, and 2 mice were selected for cell fusion. 2 MABs were obtained from the 2 mice, namely 2B6 and 2D9. The IC50 for OXZ and MPOXZ were 1.42, 0.95 ng, and 0.66, 0.21 ng. The IC50 values and cross-reactivity of the MAB 2D9 evaluated with OXZ is derivative, and nephrodizome, the cross-reactivity were obtained for other compounds. And we added different concentration to the chicken, and we can obtain the CV value, which is lower than 8.3%. The recovery rate of OXZ in chicken was 84.5 to 96.9%. And 4 monoclonal antibodies were prepared against the 4 target metabolites, and it is expected to directly detect the metabolites without a derivatization, and the IC50 value of metabolites was 0.91, 0.25, 0.8, and 0.66 ng. And the IC50 values of derivatives were 0.72, 0.1, 0.87, and 0.21. And we published our articles on the top journal, and we also applied for 4 patents. This is my presentation today. I come from the team of China Agricultural University, and I'm engaged in the rapid detection of food safety. I hope to have further exchanges with you. Thank you, Professor Yu. Due to time limit, we will enter the next presentation. And topic 3 and topic 4 have been finished. And for next speech is topic 1. And our next speaker is from China Light Food Inspection and Certification Company Limited, Mr. Fan Shuangxi. He has recorded his presentation. Now let's watch the video. 大家
First, I would like to thank the host introduction. Thanks Professor Wu and Professor Chris for the great effort on EU-China CIF project. Organize such a great online seminar. My name is Fan Shuangxi. I'm from National Center for International Research on Food Authenticity Technology. My topic is about research and application of wine authenticity identification technology with the support of EU-China CIF project. The first part, I would like to show the background. International Wine and Spirits Research Institute, IWSR, predict China will become the second largest wine market in the world, and the sales in China will increase to 23 billion by 2021. China is a huge market for imported wines. However, China is facing the threat of fake wine products. Fake wine events were reported frequently, such as ethanol mixed with saccharism, essence, pigment, water, no grape juice in wine. Wine with water addition, domestic wine is falsely labeled as high quality imported wines, and so on. The wine industry needs further regulation by the government. When it comes to the regulation, the first we need to know about China wine standards. China established the industry standard of wine and its test method in 1984, formulated the first national wine standard in 1994, revised and formed the national standard of wine by referring to a wine standard method in 2006, and defined the concept of wine of origin, vintage, and variety for the first time. From 2006 to 2010, we have wine, fruit wine analytical method, banking, ice wine, good hygiene practice for wine industry. Now we are drafting guidelines for the identification of varietal regional wines, detection of water addition in wine. It is expected that it will be officially released and implemented in future. Compared with the EU, there is still a serious lack of technical standards for wine authenticity identification in China. The EU has established a good technical system and a database for wine authenticity analysis. My topic is from EU-China CIF project WP2. WP2 aims to introduce the EU's advanced wine supervision technology system, carries out the development of China's wine technical standard, and test the authenticity of EU wine samples sold in China. GRC and BFR is responsible for WP2, and the Chinese partners are our institute, Hong Kong Polytechnic University, and Ningbo Customs. After full discussion, the project team used to IMS, GC, MAS, HPLC, and MAR to identify wine watering, topization, vintage, origin, and grape variety mislabeling, fruit juice addition, technical glycol addition, and unrolling adulteration. Next, I would like to talk about the wine method investigation. BFR uses 6 OIV standard method and 1 non-target method. We use 4 OIV method and 2 Chinese standard method and the same non-target method for watering, topization, origin identification, the carbon-13 and carbon-12 ratio values in wine ethanol were determined while we use the Chinese standard method QBT. QBT of the ethanol in baijiu that was established by our institute for the oxygenating ratio of wine water was determined. We use the Chinese standard method QBT. For the wine watering topization and origin target, the deuterium hydrogen ratio of wine isotope were determined by site-specific natural isotope fractalization, nuclear magnetic resonance sniff NMR according to the OY method. For technical gradual addition purpose, the analysis of the byproducts of technical gradual in wire CMPD, CYCDS was conducted by GC mass according to the OY method for grape variety mislabeling and fruit juice addition. Target determination of the isocyanate pattern by HPLC analysis was performed according to the OY method. For peanut grape mislabeling, determination of steamic acid wine by HPLC UV analysis was performed according to OY method. For unknown fraud, we use the same non-target wine method by proton NMR fingerprinting method, which was published by Goldman in 2013, which is commonized as wine screener. 50 bottles of the European wines were purchased in duplicate in China in autumn in 2018 from various sources such as supermarket convenience stores, specialized stores, and e-commerce by independent third-party organization. The samples many originated from France and mostly were red or rose wines, roughly reflecting current Chinese customers' performance. Vintage ranged from 2009 to 2017, with 60% harvest between 2014 and 2016. With the purchase price range between 12 RMB to 238 RMB per bottle, median at 98 RMB, about 12 euros. A rather customer-friendly wine category was adjusted. We can see the wine method verification results: steamic acid, anthocyanins, CYCDS, CMPD, oxygen 18, and carbon 13. And the determination of the oxygen 18 isotope ratio of wine water by IMS for the FID PDS. So we use the Chinese standard. So our result is also very good. For the carbon 13 and carbon 12 isotope ratio of wine ethanol, we use the Chinese standard. Difference in the workflows of the two methods are summarized in table 1. For OY method, the ethanol distillate prepared also for sniff animal analysis. The Chinese standard didn't require a distillation pretreatment. Prepared samples were analyzed by GCC IRMS directly, taking the distillation step of the OY method into account. When analyze carbon 13 alone only, the Chinese standard takes about 3 hours compared to the established OY method. So this is the result for the sniff animal result for the BFR and BCIQ. So we can see the table, we can see the result is also very good. Now we are also developing ethanol distillation equipment for sniff animal analysis. We participate in the PDS. Our results, this score is minus 2.2, and the equipment needs to be further improved. We test the same wine samples by using the same proton NMR method. As we can see from the figure, the whole CNI, FFI, and BFR proton NMR fingerprinting spectra of wine do not have significant difference. Both the BFR and the CNI, FFI achieved good results. Thanks for Dr. Teresa and Dr. Carsten. Great effort for the joint publication titled "What's in a Wine: A Spot Check of the Integrity of European Wines Sold in China." Based on isocyanate comparison, stable isotope glycol impurity analysis. This paper talks about the stable isotope ratio of deuterium, hydrogen, oxygen 18, and carbon 13, and contents of technical glycol byproduct, and isocyanate composition evaluate the authenticity of 50 European labeling wines. The test result found more than 20% of the test samples are not normal. This result strongly suggests watering 10%, topization 10%, technical glycol addition 16%, non-witness isocyanate source 12%. We are also organized several international forums on food authenticity to disseminate our achievements. Thanks for the EU-China Safe project support, we achieved several awards in the field of the wine authenticity and interest. Finally, I will wrap up my speech by showing the slogan of our center: better food, better life. I truly hope we can have such a beautiful world. Thanks a lot. If you have any questions or problems, you can send email to me. Thank you.
我们感谢樊博士的报告。
Thank you, Dr. Fan. We will have the last speaker in the morning. This is Mr. Wu Qiang from Yangtze Delta Region Institute of Tsinghua University, Zhejiang. I will talk about the application of blockchain technology in pork production. So, Professor Wu. Good morning, dear experts. I'm from Yangtze River Delta Region Institute of Tsinghua University. Today, I will walk you through the work we've done in blockchain, especially application of blockchain technology in pork production. I cover 3 topics on my agenda. First of all, pork safety traceability. Number 2, blockchain and pork safety traceability. Number 3, high credible pork processing chain traceability information management system. We made a system. First of all, this is about pork safety traceability. For pork, I have 2 parts. Yeah, I have the English version here. For pork, this starts from the rearing to processing, storage, distribution, until retail stores to the dinner tables. There are many links. There are husbandry, production, consumer. So there is management of pigs and pork, but also information. So we need to ensure such traceability. However, for safety of meat, there are producers and consumers. For producers, they need to be part of the supply chain management, as we said earlier. For a farm or for processing plant or for cold warehouse or logistic provider, for any consumer to know the information of such meat, we can scan a QR code with the phone. We know what is the source, and if we have the full chain info shown to me as a consumer, I would be able to know the transparency, and I feel rest assured because this is about safety, and I feel okay. And for government regulatory bodies, they would know the whole process and they can monitor the whole process. Suppose there were some issues, we can go back to the source. We can have the full chain traceable. As a matter of fact, for food traceability or pork traceability, before we had internet, if you would like to do it, the previous information system was in tandem. It's 1 to N in relationship. The production information, like husbandry, farm, since pigs to slaughterhouses, and they string the information data points. But with internet, with cloud computing, big data, AI, blockchain, the data collection can be parallel. Many stakeholders can collect data at the same time, owing to the features of the network. Furthermore, for food safety, it was a closed system, but now it's an open system. Previously, before there was big data or internet, in general, when we collected information, it was simple and it was a standardized way for collecting information. Before we had big data, there were experiments, and it's about one sample, and sample data are used. And there is human judgment about the sample results. But if there is big data, it's not about sample only. It could be data for every part. Then you don't have humans who are able to analyze the big data. We need to have AI. We do not need a specific result. We can look at the trends with big data. With internet, cloud computing, big data, AI, blockchain, data collection is made easier and it's made more detailed. It's not simple, standardized. Now you can tailor-make the collection of information. You can collect as much as possible, but we need to change our way of management. Same story for the slaughterhouse for pigs. We need to solve issues for them. I will share what we did working with slaughterhouse and the processing plants. We've had discussions about R&D with them, and we've got some takeaways. For the cold chain of pork, this is a common chain. This is English version. So in the beginning, there is manufacturer, producer, then it goes to the wholesaler with the primary storehouse, then into the retailer store, then consumer side. So this is a cold chain, and consumers with the product. If there is for information management, there should be a QR code with an app or a WeChat. You could scan the QR code and you know the whole story about this piece of pork. You know this process. From above, you could see this is about supply chain management. This is unidirectional. It starts from the source, either the farm of pigs or the slaughterhouse or the manufacturer, cold warehouse, wholesaler. This is unidirectional flow of materials in the supply chain. In the lower part, this is about information. For the manufacturer side, so I know where is the husbandry farm. I know it went to some cold storage warehouse. From that warehouse, it is sent to a certain level 1 or level 2 dealer and until it reaches the retailer. There is two-way information. So for the consumer with info, you could go up. For the manufacturer with such information, if a certain batch has any contamination issue, government regulatory agencies or the manufacturers could know that batch of products have reached certain supermarkets, convenience stores. For example, for the retailer outlet, they will know who this was sold to. Of course, suppose information is collected fully, whether it's a recall or whether it's accountability, it's made easier if all info is collected. There could be two-way traceability. For traceability, we have IoT, we have sensors, and we'd like to ensure this is all automated. There is RFID label or tag. For RFID tags, a batch of pork is put into a container. You don't do the human sorting if there is an RFID tag, whether it is incoming receipt or outgoing delivery. There is a sensor. It goes into the door, out of the door. All information is collected automatically. It's put into the server. You don't do it manually. That's the ideal way for management. And when we do research, we use the ideal scenario. That's the way to go. When we talk with the manufacturer, we need to look at the real case until we reach a certain user scenario. We have the ideal one as the ultimate goal, but for a specific manufacturer of pork, this is too much investment, requires too much staff, or they have to increase the price of the pork, etc. And sometimes they don't want to buy this set of equipment. So this is something we talk with them. So on the left side is data collection. There is a warehouse. We can use RFID to collect information. It could detect temperature in an ongoing basis. This is sent to level 1 dealer, level 2 dealer. What is the temperature keeping? And what is the shelf they put that the pork? This can be made feasible. Of course, you have to consider costs. On the right side is about traceability. All info is collected and that is made available to consumer as long as the consumer needs it. You don't give everything to the consumer. So there was a pickup at 9 in the morning, there was arrival at 11, and what is the temperature trending during the shift? You don't need to show everything. So where is produced, what is the slaughterhouse, etc.
So which information needs to be known by the consumers? We need to provide such information for them. So we need to collect the data as much as possible. And if when necessary, we can get such information to provide such information to consumers or regulators. This is the English version of this slide. And for pork, and in the second part, I mentioned the blockchain technology can detect the counterfeit products. From the production perspective, we focus on the supply chain, and from the consumer perspective, we provide traceability. And for food with traceability, such as pork, actually their price will be higher than the ordinary products, so that the manufacturers will be motivated to produce the food with traceability. The second part is about the blockchain and the pork safety traceability, why we should use the blockchain technology. And we also have traditional methods, traditional internet methods. What is the difference between blockchain and the traditional methods? What is blockchain? It is a decentralized, distributed technology database. It is a consensus mechanism, and it has smart contracts and a P2P network. DAPP means decentralized APP. What does it mean? All such characteristics target at the traditional centralized database. All the banks are using centralized databases. But the blockchain uses a decentralized database, and they are distributed in different organizations, different structures. And it is P2P network. And if it is centralized, then it should have a brain, but blockchain is decentralized. It has several points. So how to make decision, how to store the data, they rely on the smart contract and consensus mechanism. They have some contract or agreement, and they discuss with each other and sign a smart contract so that they reach an agreement to store the data. Through the agreement, they reach a consensus, and it is a P2P network. It is decentralized, and it is encryption algorithm. Each administrator can see the data, but they cannot get access to that. Only the data owners can get access to the data. For blockchain, it has different nodes, and the data are stored in different nodes. Different stakeholders can sign contract and reach agreement. Only all the parties reach agreement can they alter the data. So normally, it is not easy to alter the data. It's impossible to alter the data. The human costs and economic costs are far higher than the traditional methods to alter the data, to change the data. And the parties reach agreement through smart contracts. It's impossible for anyone, any party to change the data. And for blockchain, there are three categories. One is the public chain, which is readable for anyone in the world. One typical example of the public chain is the Bitcoin, which is not recognized by the Chinese government. And it consumes a large amount of power. So it has not been recognized by the Chinese government. Now there is a power shortage in Zhejiang province, which has a great impact on the private economy. And now in factories, the workers work for 3 days and take a break for 4 days a week. And for public chain, the most typical example is the Bitcoin. However, Bitcoin is not accepted or recognized by the government. The public chain has very slow speed and low efficiency. And the second category is private chain. And it means that actually there is a center to organize and manage that chain, and actually it is very similar to the centralized method. And the third category is the consortium blockchain, which is between the public chain and the private chain. And several organizations jointly maintain a consortium blockchain through smart contract. The different parties can reach agreements, and they can be permanently reserved, and the data can be difficult to be altered. So this guarantees the traceability of data. The traceability needs the involvement of the core organizations, such as the regulator, manufacturer, producer, or a third party. And they put the data in different nodes of the blockchain and form a consortium. So from the technology perspective, the data on the consortium chain are very safe. They are difficult to be altered. And the consensus mechanism does not require proof of workload like mining. The consortium chain only collects data but does not need the mining work. And the data on the consortium chain can be hardly changed or altered. So the pork traceability system is very suitable for the use of blockchain, the alliance chain. The core data cannot be altered through the smart contract. Here I would like to talk about the next part, just now we discussed the traceability, and actually traceability for pork can also be realized without blockchain technology. But why we still use the blockchain technology? Smart contract and the difficulty in altering the data are the characteristics and advantages of the blockchain technology. And there is also a concept of a timestamp in the blockchain technology. So for each product, there is a timestamp. In our daily life, when we send a message in the WeChat or the microblog, there is a time displayed. And for the blockchain traceability system, there is a time stamp. So the blockchain technology is the most suitable for the data collection of the supply chain. We all know that the information traceability can use the traditional method, but from a long-term perspective, blockchain technology is more suitable for the information traceability. And next, let's see the final part, the highly credible pork processing chain traceability information management system. This is a rather ideal solution. On the top part, we can see the cold chain basic structure, and we have for blockchain anti-counterfeiting and the cold chain industry analysis. And in the middle, we have a systematic engineering. We have smart scheduling for the production. And the dispatch. The whole process is completed with AI, and we also have internet technology. So for the warehousing, storage, packaging, transportation, categorization, and sales, for the whole process of the product, we use our technology and the device for traceability. And when we communicate with the pork manufacturers, we have a communication process.
For this traceability, before we had the system, a middle one for agricultural produce. There was vegetables. So it's a greenhouse where they produce strawberries, and we established traceability. It was easy, but plot that is made into one batch, and strawberries were picked up batch by batch, and it was sent to the supermarket. This is much easier than pork. And we talked with the manufacturers, slaughterhouses. You see, for pork, in the very beginning, there is a husbandry farm. There's slaughterhouse. Then it was sent to the manufacturer side. Manufacturer will cut them into pieces, like the big head, forearm, loin, etc. The manufacturer side have steak and all different parts are put into different batches. And for a pig, they can make that into 200 types for higher prices. They sell them at different parts. So in an ideal way, we put them into the feeder pigs and loin, which will know that piece of steak is related to which pig. If we do that in such a way, we need to collect a lot of information, and the consumer will have the information after scanning the QR code. And how to achieve that, we'll show you how this is carried out. Then, as the farm, this is a manufacturer. It could be the farm, it could be a manufacturer. We are having surveys and we talk with manufacturers a lot. It could be a cold warehouse or a logistics supplier. Information is collected at the right time. It could be about temperature, what action you will take. There are some data fields you could define. Then all info is collected about that piece of pork. Some are automatic, some are manual. So it could be both. We can use RFID in automatic collection, but in cold chain, RFID tag is easy to make, but in processing, it's difficult. You have to do it manually. After production, consumers can use WeChat to scan this QR code. You will know all info about that piece of pork. What is the pig? What is the time of slaughtering? And who cut them into pieces? Transportation, and also to a certain retailer. By using the QR code, you get everything. For this QR code, we can use blockchain to deal with any fraudulent behavior. After scanning this code, you will know that this is authentic pork. After scanning the QR code, so the instance of scanning can be passed back, and the manufacturer will know, okay, that piece of pork of mine has been scanned by consumer, then a consumer bought it, or if it's scanned in another region where you don't want to be sold, then you can scan that and get information. At different stages, at different statuses, as a manufacturer, you could have a tailor-made following about the data. And such information can be made public. And if this is an integrated supplier, you are both managing the farm and slaughterhouse and also manufacturer side, then you can have everything. Lastly, let me share what we have taken away after communicating with the manufacturers. We talked about blockchain. We talked about advantages of blockchain versus the centralized databases. This is decentralized, it's ultra safe, it's not easy to tamper with. There are advantages like these. Compared with the centralized databases, of course, this has its disadvantages. Speed of access and reading is low volume for storage, But as blockchain is developing, the government encourages the use of blockchain. As technology is being pushed forward, bottlenecks can be addressed. But that's the current situation. I mentioned earlier, all banks in China do not use blockchain. It's way too small. If you use blockchain versus central database, it's 100-fold slower. It's about access and reading speed. For Bitcoin, for one transaction to be completed, you need 5 or 10 minutes. For a bank, they have such high volumes, they don't want to use blockchain or Bitcoin. So the banks need to ensure safety for sure. Then if they can achieve safety, they would like to ensure high speed. How to deal with such an issue on our side in blockchain? For our database, we have 2 types. For pork, we use blockchain on the one hand for the sensitive data that need to be traceable. The whole process, the management of supply chain, that's part of the blockchain. But for some other non-sensitive info, like info about the enterprise, info about the operator, or the financials, so the pay grade, or after processing this piece of pork, the operator got a certain amount of salary. That is put into the central database, not on the blockchain. So it's a dual way to deal with this problem. Furthermore, when we talked with the manufacturers of pork, the ideal status is something we have envisioned. There is husbandry, farm, slaughterhouse. Then in the manufacturer side, we cut them into pieces: head, loin, steak, feet, lean meat, etc. For an operator, they could do it very quickly. Because of COVID-19, I've been to a factory once only. And on some other occasions, we talk with them through office. You see, this is operators are paid by the pieces they cut. You will know this is the feet of a certain pig, this is the loin of a certain pig. But if they put the tag there, they reduce the efficiency. Factories are not happy to do that. You know, for factory, they are applying for fund from MIT of China. We are looking at ways, a compromise. You don't have to be so specific about one pig. It could be about one patch of pigs. We can use a scan code or IoT means to do this automatically in labeling. We're doing all the trials. We are communicating with the manufacturer side. You see, we want to be ideal, but we need to consider the investment makes sense economically. If I can put up the price a bit so that I can get returns. That's all from my side. We may update you in the future. Thank you all very much for listening. Great. Thank you. Thank you, Professor Wu. We'll have the last part. We'll have Q&A. There was a question online, right? In the Q&A, no? Do we have questions online? Please open the chat box. No? There are no questions from online. I saw that there was a question. Anyway, there's no question online. Do we have questions offline? Do you have any questions? It seems no. You got everything clear. We've shown what we've done in China, and we've shown many aspects. And we have counterparts in Europe who work with us. We have WP2 is for traceability technology, WP3 another one, WP4 food safety, and these are about technologies. We have WP5 is info about consumer side, and this is about communication, and IR 2020. Of course, we have Ministry of Science and Technology and also counterpart organizations from EU. We need to show what we have achieved after getting the grants. And we need to showcase what we have done. The Chinese programs are almost completed and we're assessing the outcomes now. For the European side, they plan to do this as well. And this is delayed by half a year, and they will showcase their results in March next year. In the morning, we've exchanged what we have done in China side. And also, besides, we talked about IT technology, blockchain, how to better leverage such in traceability. And we talked about digital DNA and digital technology, etc. Then we have technology introduced and technology brought overseas, and we had two cases. One is per color rate and color rate that is related to WP, and that is related to IRR 2020 as well. NMPA have reviewed such drafts, and there are subcommittees for physical and chemical committees. And after review, NMPA will list such methods, and the NMPA will be soliciting feedback from users. And you are free to give us feedback. In general, the work we carried out on China's side has come to an end, and also it's become a typical example in MOST.
And the Technology Assessment Center of MOST has finished the evaluation and submitted the assessment result to MOST. And in this process, we implement one thing, that is international collaboration is very important. In our collaboration, we needed to introduce some technologies. And our collaboration should be two-way. We not only introduce technology from other countries, but also contribute China's wisdom to other countries. in international collaboration, we should focus on solving our problems. And we should also introduce the advanced technologies from other countries and also export the technologies from China to other countries. This morning, the experts from China presented their research results. And in the afternoon, we have for the M session, and we have speakers from Europe and speakers from Queen's University. Mr. Chris Anion will be the moderator for the afternoon session, introducing the work and experience on the European side. Please come to our conference hall at 2 p.m. Let's listen to the presentations from the speakers from Europe. We have quite a lot of experts online participate in our conference online. And okay, thank you. This is the end of the morning session.